Welcome to Quantitative Seminar this week. I'm Zoe. I'm one of the organizers this year, and I'm going to start us off with a land acknowledgement. So the University of Washington acknowledges the Coast Salish peoples of this land, the land which touches the shared waters of all bands and tribes of the Suquamish, Tulalip, and Muckleshoot nations. Our lab also acknowledges that we live and work on the ancestral lands of the Jawamish people, the first people of Seattle. I'm really excited to introduce our speaker today. Our speaker is Alexa Fredston. She is an assistant professor of ocean sciences at the University of California in Santa Cruz. She holds a PhD from the University of California, Santa Barbara, and graduated summa cum laude from Princeton University. Previously, Dr. Fredston was a postdoctoral associate at Rutgers University and a High Meadows Fellow at the Environmental Defense Fund. So Alexa is happy to take questions as you go. So feel free to just shout out questions, raise your hand, and I'll be reading the online questions. And thanks everyone for coming, and a big thank you to Alexa.
Thanks, everyone. I'm going to emerge from this little corner and stand over here, so hopefully I won't block the slides, but you can all see me. Thank you for letting me crash your quantitative seminars. I'm very excited to be joining you in this California-like weather, which I assume is what it's like all the time in Seattle. I just moved to Santa Cruz, and I'm happy to be back on the West Coast and re-engaging with people on the West Coast about ocean stuff. So this is a somewhat casual kind of romp through three main research directions that I've been working on, and you're welcome to chime in at any point, ask questions, and if we don't get through all of it, that's okay. I frame this kind of around the idea of prediction and forecasting. So I'm going to start by giving my extremely qualitative and subjective opinion of how we're doing on this topic, which I was thinking about earlier this week while I was getting ready for this talk. So I think it's helpful to think about how well we're doing at predicting marine ecological responses to climate change, sort of separately about how we predict them over space and how we predict them over time. We have a lot of studies predicting climate stuff for climate responses over space, and I'll show you a few that will kind of build up to where I think we need to go. This is a study that's 10 years old that some of you have probably seen, looking at marine fish, showing that their position in at least latitudinal space, which is on the y-axis here, is very well predicted by temperature, and specifically where temperatures shift in the oceans. What I mean by that is if this fish historically was found at, I don't know, mean annual sea surface temperature of 12 degrees Celsius. If you track those 12 degrees Celsius through the oceans as they warmed, did the fish go in the same direction at the same rate? And this plot shows that generally they have. That's from bottom trawl surveys in North America. This is some of my own work using the same surveys to look at the edges of species distributions. These are poleward or cold range edges in the northeastern US. And the dashed line is perfect climate tracking. So it's the equivalent of the red line on the left plot. If they sit on that dashed line, they went exactly where we predict them to go based on temperature alone. Most of them don't do that, but they do kind of cluster around that line, and it's much more likely that they shifted north, which is to the right of the solid line, than that they shifted south during a 50-year period of rapid warming. And this last plot is from kind of a different approach to thinking about spatial prediction in the context of climate change. This is a study on mangroves that looked at how mangrove density changed from year to year. That's on the y-axis, and temperature on the x-axis. And the authors were hypothesizing that there's kind of this break point where when it got cold in the previous winter, all the mangroves died, and that's what sets the mangrove rainfall. What I want to draw your attention to in all these plots, though, these are all very statistically significant results, but they all also have a huge amount of variability if you were to look at a particular dot. So here you have a bunch of species that shift a lot, even though they're not predicted to at all based on temperature change. And you also have a bunch of species that kind of don't shift at all, even though it warmed a lot. Similarly, in this one, none of these dots are on the perfect tracking line. Some species shifted south, we have no idea why. And a lot of them didn't shift during a period of rapid warming. And here you have some mangroves that did better when it was freezing, some that did just fine. And then you also have some that died off for reasons that weren't related to temperature. And that highlights the challenge of moving from correlations to actual forecasts of the future. If you wanted to predict any one of these dots, this is not enough. And the same, I think, is true when we look at one place over time. So this is a cool study looking at the Barents Sea and the community turnover for more warm affiliated species. So the blue dots are Arctic species, the red dots are lower latitude species. And you see in the almost decade that they studied it from left to right, that in the points where they did the straw survey, there's a turnover toward warmer affiliated species. But again, a lot of variability. Could you, for any one of these dots, predict what was going to happen? Maybe or maybe not. This is a personal favorite. This is from Monterey and Hopkins Marine Station, where a group of researchers discovered a survey of the intertidal from 1932 and repeated the same survey in the 1990s and found a huge increase in southern affiliated warm intertidal species and a huge decline in northern affiliated ones relative to that baseline 6 years prior. This is a great study, but it's also the case that in general in the literature, when you have studies with only 2 time points, an old one and a new one, it's pretty hard to infer a lot about the intervening change in just those 2 time points. This, some of you may have seen, is from a big synthesis of the impacts of the blob in, I don't remember if this was Gulf of Alaska or the whole Northeast Pacific, but someone here might. The black lines are the adult fish. The gray band is the heat wave. Obviously, you can do the math. The larvae, which are in red, did great. All the adults declined. So this is an extreme event, a different form of detecting change in response to the environment. But again, when you look at the scatter of the data, a lot of what I've been focusing on the past few years is how we get from these broad brush patterns to predicting individual changes. And these types of approaches may not get us all the way there. So the question then is, like, if we have these really strong statistical things showing that things are moving in space, showing that there's community turnover for warm affiliated species, can we actually make predictions? And I think in a lot of cases, we need more information and more understanding of why things are changing to be able to forecast the future. This is obviously not just my idea, and a lot of people have made this point. I think the state of the literature is sort of a combination of maybe a bias towards publishing positive results and not publishing really cool studies like this one, which looked at an intertidal community after 50 years of warming and found absolutely nothing. Those are harder to publish, speaking from personal experience. And also just enormous heterogeneity in how individual species respond to the same environmental change that we still don't fully understand. So the theory question that I work on is why species are found where they are and not elsewhere, and what makes them move. And to kind of make that more precise in the context of applying it to oceans and ocean forecasting, I have been thinking a lot about how well we can forecast range shifts and the ecological effects of climate change from process and from the environment. So I've been doing this sort of across scales, and I'll argue that at the scale of populations, we still don't have a super clear sense, if you look within a species, of why individual populations respond the way they do to environmental change, and then how that aggregates up to the species throughout its range. We also still, I think, have a lot to learn about what sets the species range itself, why the edges are where they are, what makes them move. And so without that, maybe not a huge surprise that it can, at the moment, be challenging to predict how communities are going to respond to environmental change. So I have kind of divided the talk in these 3 scales. So I'll start with, I'll start with the range edge question, delve into population modeling, and then scale out to marine heat waves and fish communities at the end. I like studying and talking about range edges because everybody knows what they are. It's a you know it when you see it thing. This is a range edge. We all know it. We've all seen tree lines. And a tree line lets you imagine very easily the question of why range edges are so cool, which is, why is this specifically here? Why isn't it further up the mountain? Why isn't it further down the mountain? If you stood there and looked along the tree line, what's different about what's on the left versus what's on the right? That's kind of the question of range edges, which is a super old question in ecology. This is something that Alfred Russel Wallace, who was Darwin's contemporary, was super interested in. And it's one where we have like a ton of theories and hypotheses, very few tests of them, especially in the oceans. Most tests of range edges are elevational because it's easier to study elevational range edges on land. So we don't have a lot of theory for predicting why these things fall where they do in the oceans. And my PhD work was trying to actually go out and field test some of those theories with the bottom trawl surveys. So I'll show you one of those projects, which was focused on this thing that is called the ecophysiology hypothesis. You all know what this is, even if the name doesn't sound familiar. It's just the theory that the ranges of marine ectotherms are limited by temperature, and importantly, at both edges. This comes from some really cool studies showing that when you take a marine critter in a lab and you either cook it or freeze it, and you like write down what temperatures it died at, and then you go on and look at their ranges in the oceans and look at the extreme temperatures that they inhabit, those are very similar. So the theory is maybe they actually really are living up against their absolute physiological limits. And the corollary of that is that at both the cold edge, which is also the polar edge or the leading edge if the thing is shifting north, and at the warm edge or the equator or trailing edge, it should move as soon as temperature changes with almost no warning. This assumption is actually baked into a lot of models of ecological response to global change, but we have no idea if it's true. So we set out to test this with 3 of the bottom trawl surveys, the West Coast one, Eastern Bering Sea one, and the Northeastern one, from which I calculated 165 range edges. Most species in any given survey don't have a range edge in the region. Their range extends way beyond the bottom trawl survey on both sides. So that shortens the list of things you can study. There's something else I wanted to say here. And generally the approach was just to predict where the edge was every year based on the bottom trawl survey data from NOAA, and then ask at what temperature was the range edge found every year, and is that temperature conserved over time? So we estimated the range edges with VAST, which was developed by Jim, who's here. This is not my plot. This is a much better plot than what's in my paper of how VAST works. So VAST estimates kind of a spatial temporal density field for the species from which we extracted range edge positions. Something it has a number of advantages, including for dealing with some forms of the survey variability. It allowed us to combine the 2 West Coast surveys, the older and more recent one. And it also allowed us to calculate a standard error around the range edge position based on the variation in the estimate. So this just generally gives you a sense of the type of data that comes out of VAST. And we did this in these 3 regions that had very different warming histories to see whether we saw different patterns of temperature tracking in places with really different climates. The Northeast has warmed very significantly and consistently. So summer temperature, which is the red line, winter temperature, which is the blue line, and mean annual temperature, which is the orange line. At least the top 2 have warmed more than 1 degree in 50 years, which makes it one of the fastest warming places on Earth. The West Coast, as we know, during this interval for which we have trawl data, which you can see on the x-axis, did not actually warm, but it did have huge changes in temperature related to oceanographic phenomena. And then the Eastern Bering Sea warmed, but non-linearly through these kind of huge warm, cold events. So the question is, in these very divergent climates, where and how much do we see temperature tracking at range edges? So the plot I'm going to show you tests that for both summer and winter temperatures as predictors. We generally think in biogeography that extreme temperatures should predict ranges more than means, but we have no idea which extreme. So it could be that the range edge is limited by how cold it gets in the winter, or it could be that it's limited by how warm it gets in the summer. So we tested both. And on the x-axis, like some of those plots I started with, to the right is a positive shift along the coast or north, and left is a shift south or towards the equator. And then on the y-axis, if something was perfectly tracking temperature, you would see it on this horizontal 0 line, because the temperature at which the range edge was found would be the same every year. Here are the actual data, and they're kind of colored and shaped by whether it's a poleward or equatorward range edge and which region it's in. So there are 3 kind of take-home messages from this paper. The first is that most of them do overlap with this 0 line. So at first glance, this seems like fairly strong evidence that temperature predicts the position of marine range edges over time, not just kind of in an evolutionary biogeographical sense. The second thing, and this is not rocket science, is that things that moved really far north went someplace colder, and things that moved really far south went someplace warmer. So the departures from climate tracking kind of happen when things moved great distances. But the problematic result is that most of the outliers that did not track temperature particularly well were in the Northeast, the place that's had 10 years, or sorry, 50 years of very consistent warming. Which I worry means that the reason these others track temperature is more because, for example, here on the West Coast, if temperature didn't change that much over 30, 40 years, which we have data, and the range just kind of also bounced around. With this approach, that looks like temperature tracking, when in fact, it may just be that the species didn't move that much and temperature didn't change that much. So I would have been more convinced that this demonstrates that the ecophysiology hypothesis is predictive, had the place with a ton of warming still had its species demonstrate that they conserve their thermal niches, which they, by and large, actually did not. So this summarizes what we just talked about, that these niches were least conserved in the Northeast, and that obviously is a problem for management and forecasting. I normally start these talks explaining what fisheries management is and why prediction is useful. I kind of skipped over that for this audience, but you can certainly imagine that it would be helpful to have a better sense of where stuff is moving and how its abundance is changing, which currently is a little bit challenging with models that mostly use temperature.
How do you deal with firm boundaries like land? I mean, is the Northeast like that because you don't have the range edge for the colder and farther north?
Yeah, great question. So we calculated the range edges going along the coastline. They do run out of the survey at some point, and the North run out of the country. So we don't have data on the far side. We didn't use edges that started out right up against the edge of the study domain, because to your point, then we can't track their dynamics. So all of them had to fall within the study domain, at least at the start of the time series. And then if they got out, we just sort of used the furthest points where they were found. Does that answer your question? Awesome. Yeah, cool.
Can you remind me how you define range edge in the VAST models? And I guess my question is, are these results, insensitive to changing that? I assume it's some like percentage.
Yeah, we use 99th and 1st percentile of biomass. We also tested the sensitivity to 5th and 95th and got basically the same results. It's a little sensitive to the method in that we get slightly different answers doing it on the raw data versus doing it with the model. I think part of the interpretation of that is that the model deals with the variability in the raw data, that he found the metric if you just calculate it on the raw data, but there's no guideline whatsoever on how to estimate range edges, something Cody and I talked about for a long time. So you just sort of have to make up your own method, which is obviously not ideal. And we don't have a great sense of the sensitivity of these methods in general. Yeah.
You consider this in a more of a community context? I mean, do their predators and prey also move in those same range?
Great question. Not in this study, and I'd be very curious too. There are these stories of this happening, of course, the North Atlantic right whale chasing its prey further north. My knowledge, most of these species are somewhat generalist predators, so it's a little bit challenging to kind of match them in that way. There are some kind of more focal studies in the system showing, for example, that with the decline of cod, a species called spiny dogfish has kind of come in and taken over its distribution and maybe shifting to track its prey. But I'm not totally sure how I would incorporate it in kind of this large scale way. You have an idea from you?
Okay.
Please.
I'll ask one follow-up question just to make sure I understand properly. So there's so many species that are kind of where their survey runs out, I suppose, in terms of where their kind of whole range edge is. So if the species kind of move up against the edge of the survey in year 10 of like the time series, does that range edge then just stay static at the kind of northern edge of the survey, where that species may actually be kind of redistributing much further north, but we're just not able to capture that with the survey? So
that's exactly right. Because we were pretty aggressive in throwing out species that started close to the edges, that didn't usually happen in like year 10 of a survey, but it might have happened in year 30 or something that they made it up that far, and then it would just show that they were stuck. Yeah, exactly. So I love working on range edges. I think they're very cool and very understudied. If anyone needs PhD projects, I think this is a great direction to go in. I've been working a little bit on in the intertidal and subtidal actually, which is a even clearer test case, I'll say, than the bottom trawl surveys, because intertidal and subtidal from a biogeography standpoint are just one-dimensional. Like things can go up the coast and down the coast, but they can't go really anywhere else. And that's a particular concern at biogeographic boundaries, which are places in landscapes and seascapes where lots of species have range edges in the same spot, suggesting that over evolutionary timescales, that was the dispersal barrier or some other barrier that made it impossible for them to establish on the other side. These exist in the oceans, but we don't have a clear sense of why species don't exist on the other side. They tend to be places both with very complex changing ocean currents and with really steep gradients in habitat and temperature. So it's not super clear. Can species cross? They can disperse across just fine. It's just maybe too cold on the far side, for example. Or can they not cross at all because ocean currents prevent them from dispersing? And a lot of intertidal and subtidal critters don't really move as adults. So we're testing this at Point Conception, which is a major biogeographic boundary in California, actually by doing eDNA surveys on both sides throughout the year and comparing them to kind of historical maps of species to see if we can detect larvae on the far side, but never adults, which suggests that it's not a dispersed barrier. And this is a project with, I put up all the names, the Zagar Family Foundation and the Nature Conservancy, which has a preserve at Point Conception. And they would like us to tell them if they're eventually going to need to like literally move the critters. So this is a fun way to think about range edges in a very applied context. So we're talking about range shifts a lot and temperature change and our species shifting when temperature changes. But one thing that we haven't really talked about yet is how this actually happens. We know it doesn't happen. They're not forward thinking. They're not like looking at climate models and deciding where to go. This is a reactive emergent property of species or populations and individuals kind of doing their thing. So when I started digging into mechanistic modeling, I think it's helpful to like put together like a story of what you think the populations are doing and then see if that bears out in the data. So one story you could think of is, okay, let's say warming causes warmer winters. That's not crazy. We know that's happened. Now let's say that because winter got warmer, the individuals in the northernmost populations do better. Also not crazy. Those are the marginal ones living someplace really cold. So if it gets warmer, they might either survive at higher rates or have higher reproductive output or both. That means that you'll get basically more density at the poleward edge because they potentially will have more offspring. And that leads to an increase in polar populations, which would either drag an estimator of the edge north or literally might mean they spread north if they start kind of spilling over into habitat. So that is kind of a story for the poleward edge. The equatorward edge, maybe the warmer winters mean it got too warm. The organisms experienced physiological stress, they couldn't survive there, and they either die or leave. That causes a retraction of the equatorward edge. And I think unpacking these mechanisms kind of for every species that we study or we know might be shifting and thinking about which ones are really reasonable is helpful for moving towards process-based modeling. I'm not the only person to talk about process-based modeling. There's kind of this general sense that the future of predictive ecology could potentially be more mechanistic. And some of this has been really focused on range shifts, but a lot of it is just generally focused on ecological forecasting and being more process-based and mechanistic, same thing, so more mechanistic. So what would a mechanistic model of range shifts look like? Well, most models of species ranges that we think about start with the blue stuff, which is where the species is potentially and some predictors, and then tries to predict the red stuff, which is distribution and abundance. So you might have the presence of another species in your model, you might have environmental variables. Process-based models just make that relationship explicit, usually using population dynamics over space and time with things in them like density dependence and population dynamics, dispersal, and so on. This is not a schematic that I made, but it summarizes kind of the goal of making the implicit explicit with this approach. And right, so the type of this that we've been using, and then I'll kind of tell you more about why it's cool, starts with writing down a model that describes how you think the world works. So that might kind of include the processes in the last slide. It might have density dependence in it. It probably has some spatial dynamics and dispersal. You might want it to be age or stage structure, depending on your interest in your application. It might have other species. And in my case, it kind of always has to be a hierarchical model that also accounts for the way the data are collected, because you're fitting to bottom shell survey data. They're not unbiased in the way they sample. But you kind of go away and write that whole thing down, and then you fit it to the data. So what's unique about this approach called dynamic range modeling is that you don't need most of the parameters up front, you just need a ton of data. And it estimates the parameters for you. You may or may not think that's better than going out and getting the parameters yourself, but some of these parameters are very difficult to measure. And with this framework, it estimates all of them from the data. Once you have the scaffold of the model and the parameter estimates from the fitting routine, you can run it forward and make a forecast. Okay, so these things are really useful for forecasting. Why don't we do them? First of all, you need to be able to write down this model, which means you need to have some educated guess for the underlying population dynamics of a species, which can be challenging if we really don't know a lot about them. These models are also super data hungry, and if you just kind of write down one, it's possible that some of your design decisions are really what's driving the results of the model, not the processes themselves. But if it works, it would be awesome. These models can be used to kind of falsify ecological hypotheses. If you write down a model with a stock recruit relationship in it, it consistently fits terribly. That might tell you something about whether there is density dependence in the population you're studying. They also are very scalable. You can have physiological mechanisms in them that affect individuals. You can also have kind of macroecological landscape, seascape scale effects. They're really flexible. If you wanted to design one that has local adaptation or fishing or human impact, or rather human user decisions in it, basically anything that you think you can parameterize can be programmed into these. And so for that reason, they're potentially useful for near-term forecasting. So I've been fitting these models for 4 species, although I'm going to just talk about summer flounder today in the Mid-Atlantic, in a partnership with the Mid-Atlantic Fishery Management Council, using the Northeast survey and fitting them to 35 years of data and then doing a retrospective forecast for 10. Retrospective forecast means that we forecast the past. The model doesn't see what happened in the intervening years, but we know, so that we can very clearly evaluate how well the model did. We just talked about this. So we tested it every year for a decade. And I'm going to walk you through a little bit of this model structure. I can go kind of fast, but you're welcome to stop if you have questions, because there's a lot of stats going on under the hood, which I won't mostly roll out, but I'm happy to explain further. So it's a spatial model, as I've been saying. It's structured with patches along the coastline, and that's actually what was in this. I should have said map. Those blue bands are the patches. In each patch, we have age structure. So it's spatially structured, it's age structured. So the fish age every year. They also die every year. And they're self-recruitment. This is almost the simplest model that you can come up with for population dynamics with age structure. And each of these gray things is a parameter. The gray arrows, that is. Of course, there has to be more than one patch. So individuals can move between them. We have a sort of various options for recruitment that are more global, or adults can move between adjacent patches. And of course, in every patch, all the same stuff is happening. In every time step, it's a discrete time model. And I think the last detail to tell you is that some of these rates are temperature dependent in the model. So that allows us to explore how temperature changes the population dynamics of these species over space and time. And we programmed it so that you can choose whether temperature affects recruitment, mortality, or adult dispersal. And obviously those represent different hypotheses about how this critter is actually really responding to temperature change in the oceans. This is basically saying the same thing, but in the actual framework of the model, which is a Bayesian hierarchical model. So the thing we actually want to estimate is in the middle of the slide, in white, the true number of fish, every patch, every age, and every year. We model it as conditional on these various parameters, which are the gray boxes. The only data that we give this model, in addition to the trawl survey records, is temperature and the fishing mortality estimate from the stock assessment. I'll talk about a little bit more later. We don't use it in all the models, but it's an option to use it. But obviously we are not lucky enough to just be able to fit this to data, because we only have a trawl survey for these species, which has enormous amount of size bias. So instead, we are actually fitting to, or rather, we're calculating this quantity both from the process model, which was on the first slide, the population dynamics that we think are going on under the hood, like recruitment and density dependence potentially. And that's calculated from the trawl vectors, which are conditional on that, in addition to conditional on the sampling process, which has things like catchability and process error. So this is a Bayesian network that 25% of people love seeing, 75% of people hate seeing. The past 2 minutes was very painful for you. We'll just move on. So this is not one model. We ended up with the full factorial design of 64. Because you can move temperature dependence around to either recruitment, dispersal mortality, or not have temperature dependence. You can turn on and off stochastic recruitment. You can turn on and off using the fishing mortality value from the stock assessment, which we need to be able to turn off for some species where we don't have it. But it's generally helpful for the models. We also have an option to fit to length data. And I put in, so Jim is part of this paper who was developed the movement model that we use for temperature dependent dispersal. Dan Ovando was also instrumental, who was here for a long time, in designing the model, especially in fitting it to length data to inform the age structure, which is a feature that you can turn on and off. Really, it doesn't always help. We thought it was going to be super helpful in figuring out the age structure to fit to length data in addition to abundance data, but sometimes a data conflict arises where the model can't parse the two, which has been interesting. And you can also turn on and off a stock recruit relationship that ties the amount of recruitment to the amount of adults. So I'll show you one of these dynamic range models.
Can I ask a question?
Yeah, please.
I guess I'm kind of trying to see, how is this different from stock assessment? It's basically explicit stock assessment.
Well, it's a really bad stock assessment. That's one way it's different.
Yeah.
It's a spatial stock assessment. It is really focused on the temperature dependency of various aspects of life history and how that influences the spatial allocation of abundance. So
Dispersal and movement and things.
yes. So if we had 10 really good stock assessments end to end and understood how those populations interact with each other, that would be better than this model. But for most, we don't. We have 1 to 2.
I guess we couldn't have, like, we would never try to do that because there's not enough data. So I guess then my question, like, how is there enough data for these models? There's not enough data for the stock assessment that is spatial.
Yeah, so I don't know enough about stock assessments to answer half of that question. I will say that we do have enough data for these things, as you'll see in a second, to have substantial predictive skill at predicting some kind of core features of population dynamics, like did the population increase or decrease? Where was the center of abundance? Which patches have most city abundance? Some of it, I think, depends on your goal. Stock assessments are trying to guess exact abundance, which is a really hard thing to guess. We have no interest in exact abundance in this project, and it's all about relative abundance in a spatial, like across the spatial gradient, basically, which is potentially an easier thing to do with the same amount of data. But not totally sure. Yeah.
Surprised, doesn't seem like you're using temperature preference. Isn't that information available, and how much is it vary between the stocks?
What do you mean by temperature preference?
Well, like, we go to the lab and see where, like, how your experiment you said before. And
Yeah.
Temperature extremes.
The hard thing, it wasn't obvious to me, but again, would love ideas on what that means in a mechanistic sense. Because if this is a population dynamic model, we estimate an optimum temperature for the different processes that can be affected by temperature. But then the question is, if you have the optimum temperature for performance of a fish in a tank, but is that also its optimal temperature for recruitment? Is that also the temperature would shift to a habitat in? And is it also the temperature, beyond which it would die? So instead, what we've done is estimate kind of one, we call it optimum temperature in the model, but it means something different for each of those because it's the temperature at which that process is optimized.
Yeah, I just started to think dispersal would be the place where you could use that information.
No. Yeah, that's true. We could inform in the movement model, we could inform that prior with the lab studies, which hadn't occurred to me. That's a great idea. These tend to be pretty easy for it to estimate because the stock is overwhelmingly distributed in the southern part of the survey, so it kind of knows that that's the temperature that it likes, but that's a great point. Yeah.
I will chime in on that, like that, I would assume that the partner database
Mhm.
that has the recruitment, like larval stage mortality sensitivity, but like,
That's true.
I'm not aware of any database that has selection experiments on movement parameters, like the behavioral ecology program is starting to set up like selection experiments on cloud. But if anybody knows of like a database
Yeah,
of
that'd be
selection
cool.
experiments or movement, that would be like a gold mine. In the state.
And I think this is part of a deeper conversation in physiological ecology about like, what do these vital rates that we measure mean in nature for species experiencing global change, which I am sort of lightly aware of, and I think is related to this question, but not something that I've delved into in the context of this model, but that's a great idea.
Does the within area stock recruit function make biological sense for these species? I know a lot of our species, west coast of Alaska, tend to have extended durations of the pelagic zone that can take them across large areas.
Yeah, that's a good point. It would be very straightforward to change the stock recruit relationship to use region-wide spawning stock biomass instead of patch level spawning stock biomass. I think that makes more sense. I can add in a toggle, although then I'll have 100 model. But that's a great point. I'm gonna write that down and please go ahead.
My question was kind of related. So you're assuming that when a fish moves from one patch to another, it
Yeah.
takes on the characteristics of that patch, right? It assimilates to that population with the mortality rates and recruitment rates.
Great question. So most of these things are, well, yeah, that's basically true. We're not literally estimating mortality in every patch in every year because we don't have enough data to do that. We're estimating the parameters of a function that make that temperature dependent, and then we just kind of back out how it affects the patch. So it's kind of one mortality rate, one preferred temperature, one temperature sensitivity, and then every patch gets a different rate. But you're right in essence. So when it moves, it gets the new properties of that patch.
And then they self-recruit within that.
Depending on, I forgot a toggle, depending on whether stochastic recruitment is turned on or off. So if it's on, recruitment is just a random global process. If it's off, that's what happens. Yeah.
Been dealing with similar issues, trying to understand that critters have moved somewhere else or died. And
I have no idea what you're talking about.
But in simulation testing some of my models, they did okay predicting changes in mortality, but really poorly in changes like catchability and movements. And I was wondering if you had simulation tested with this to see if you could estimate parameters that you're trying to estimate. Or
Dan has, and yes, but I don't remember how that differed among parameters. I'm sure there was some really better predicting than others. That's a good thing to remind myself of. More fun? More fun. Okay, I'll show you some plots and then we can keep talking. So I'm going to show you plots from this model. These models I like happily designed working from home with my homemade bread over the course of the pandemic, and then tried to run all 64 and discovered that's very challenging. So I spent the past 2 weeks battling with the high performance computer at Rutgers, which I have still not defeated. So I just ran a little toy model on my computer this week to show you. It does use the F values from the stock assessment. It does have stochastic recruitment. I have temperature affecting movement. So that's the Jim Taxis model. And I turned off the stock recruit relationship and the length thing. So let me show you kind of what we're trying to predict. This is what actually happened to summer flounder over this decade. We had the misfortune with this particular species of choosing one where in a decade when it warmed enormously in the Northwest Atlantic in 2012 and 2016 were heat wave years, the thing didn't really go anywhere and it moved south in the 2 heat wave years, according to the raw survey data. So on the y-axis, the latitude you can see is in decimal points. So it didn't move very much. The weighted range center, which is what we're plotting here, is around 38 degrees north. So this is what we're trying to forecast, which is a little bit of a challenge. And here's what the model actually says. The blue line is the model's best estimate. The bands are the probability density, so they get lighter as you encompass more of the potential parameter values. Take the bands here with a grain of salt because I only ran this for like 1,000 or 2,000 steps on my computer. The real ones will be run for much longer, and the model will develop a kind of slightly more constrained estimate of these values. It gets the centroid in the right place, and it predicts that it lightly moves north, which most of them do because it warms. And so it predicts some slight changes, in this case, in the preferable habitat. But all of the red dots do fall within the posterior distribution of the centroid estimate. The real test, though, to your point, is how this performs over space. And this is the plot, sorry, this plot's a nightmare, of abundance over time in every patch. So the gray number on the top is latitude. So the southernmost patch, you can see there was almost zero. Summer flounder, they did kind of better in the middle of the time series and then kind of went back to being low. And then you can see the black line is the model's best guess. It did not do great in this patch. And I'll also point out that the y-axes are different on all these patches. They're scaled to the abundance. So some of these are just, most of the stock is kind of in this 36 to 40 degrees north. The model gets and generally understands that it's not really present in the northernmost patches, although interestingly, the movement model overestimates how many summer flounder are in the really cold patches further north. So the other models don't do that, which has been interesting. But this one has, I think it thinks the northern patches are more suitable than they actually are.
It's estimating time varying, patch varying mortality.
It's estimating time varying, patch varying abundance. It has the fishing mortality. And it also, so abundance is affected by mortality. It's affected by,
And when you say mortality, is that just the fishing mortality?
it's a fishing and a natural mortality, which is also in the stock assessment. Thanks for asking.
And so that doesn't, and that does or doesn't vary by year patch.
It varies by year, but not by patch. We don't have spatial fishing information. So mortality is the same everywhere unless we make it temperature dependent. I would love to change that. We have not gotten spatial fishing ever. The other things that affect abundance are movement between patches, which is like random diffusion, unless it's a movement preference model, which this is. Recruitment, which here is like a global random process, so that doesn't vary that much among patches, but also the population dynamics in the patch. So obviously, if if it thinks there was a very good recruitment year several years ago, that will carry through in each patch based on the abundance.
Get thoughts on, it looks like most of your estimates are larger than your observation.
Yeah, that's not true for all the models. It's mostly true for the movement models, and I can't quite figure out why. That's something that until I run all the models, I can't totally interrogate. I'm not super worried about it because, like we were saying, the overall abundance is not super important to this project, but it is curious, especially because they don't all do that, some of them. So I'll just show you one patch where most of the density is to kind of, this is again sort of blown up by this error distribution, which I think will shrink when we run it for longer. But the model does get some of the interannual variability when it's hard to see because of the y-axis, but when the stock got more abundant, kind of early in the time series, it captured that, and it also captures its increase at the end, which is somewhat exciting given that, again, the model doesn't see any of these data, it's guessing. So to put it all together, and I showed these plots last because they don't have the uncertainty in them. This is a summary of the abundance over space and time in the true data. So every vertical stripe is a year. If you're flying over the east coast of the US looking down, the y-axis is latitude, and the bright spots are where the fish are. So some of the features here that you'd want to reproduce in the model is that there's overall increase in abundance over time, but the abundance actually mostly stayed in the center of the distribution, and that there were kind of these particular patches that did well in particular years. It actually gets both those. It gets the increase in abundance over time, and that is partly the fishing effect. It also gets that the density is generally in these areas. This particular model puts too much density far south and far north, which they don't all do. But generally, most of these models, or at least the better dynamic range models, seem to substantially be outperforming the null models that we have. So we're also in the process of testing these against more correlative species distribution models and each other once we run all of them. But they do seem to have some skill for short-term forecasting, and part of our conversation with the Mid-Atlantic Council has been exactly what do you want to forecast? Because there's some things that these clearly are not great at forecasting, but other things that they are pretty useful for. So one thing that we've talked to them about is understanding what proportion of the stock is above and below the edge of their management domain, essentially. So basically what's out of the mid-Atlantic bight. That's something they're really curious about. And that's something that this model is actually very well set up to do. So I think we've asked them to sort of think, and then we'll check in with them in a few months about what types of metrics and then what types of associated uncertainty do they most want to hear from this kind of model, which obviously is not yet a future forecast. It is a retrospective forecast. But most of the best models have a fishing effect, that's not a surprise, and stochastic recruitment. And I think there probably won't be one best model. So the movement models tend to get the up and down along the coast pretty well, but don't always get the overall stock distribution as well as some of the other models, which is something that we're going to parse when we get all of them back from the computer. So I think these models are super fun and I'm excited to keep working on them. One thing that we've talked about doing out here in the California current, this is, by the way, what it looks like when you run the model for longer and you have a slightly more confident estimate, although this model is very wrong, confident but wrong, is trying to sort of calibrate put these in the California current, which has a lot of like fairly high performing near-term oceanographic forecasting data products, and then be able to make some actual future forecasts with the associated, hopefully correctly quantified uncertainty. But in general, oh, sorry.
Question from online.
Yeah. Hello,
What
online.
about competitors and predators whose distributions are also shifting?
I'm so glad you asked. That's not in these models. I would love to. So like I said, you can put anything in these models. Hopefully you're getting the sense for this kind of like simulated toy world, so long as you can parameterize the model. I think predator-prey dynamics and competition are an obvious thing to include, but you'd have to kind of have information on both distributions. That's the easy part, but also understand how they interact with each other, how to kind of understand what those parameter values should be, and give the model enough data to basically then it would have a new source of mortality if it was a predator-prey relationship, for example, and have it be able to differentiate that. So the challenge is always the data for the parameter estimation, but given that, I do think there are lots of cool directions to take these in, and this model is written in a Bayesian language called Stan. If anyone would like it, please tell me. Would love other people to use it. And I think it's relatively straightforward to adapt to different use cases and different data sources now that the kind of hierarchical Bayesian structure exists, especially for bottom 12 survey data, because that's what it was set up for. Okay. In the last few minutes, we've gotten to the fun and easy part of the talk, which is marine heat waves. So, and now to remind you, we're kind of zooming out to the level of communities. I think this is what a lot of people, not necessarily here, but in general, think of when you hear marine heat wave is like this, is a horribly bleached coral reef and a lot of ecological degradation. But this is sadly not where I work. I work on stuff like this, which is way less glamorous, and it's way less clear what the heat wave impacts will be in these types of systems. So we've been studying this, and as with all of this, I kind of beforehand tried to figure out like what are our predictions from what we know about these species and communities, and then can we develop a test with the data that we have. So I think what a lot of us picture happening after heat waves is this: you have some community that's made up of cold and warm affiliated species. Then there's a heat wave and you lose the cold affiliated species, so you have a decline in biomass or abundance or whatever you're thinking about in your community. And that could happen, but there's other stuff that could happen. It could also be the case you gain warm affiliated species in a process called tropicalization. So you don't actually have any change in biomass, you just have a change in community composition. Maybe that happens and you don't lose the cold species, you actually gain biomass. Or maybe neither one of those happens predictably. These are obviously testable hypotheses about changes in biomass and community composition following marine heat waves, which we've been working on testing with a really awesome data set from all of the publicly available government trawl surveys around the globe, which are on this map. There are 18 that we used, and we calculated marine heat waves within each one. So we, people who may have thought about marine heat waves usually define them as like a percentile anomaly of temperature. So if you say a marine heat wave is when temperature exceeds 90% of what it historically was, that means 10% of the time you'll have heat waves. This was done with a 95% threshold, so that means 5% of the time in every region there was a heat wave. And those lines show you when the heat waves were. Obviously, some familiar regions on this map. And I want to give a quick plug. This map is a product from a working group called Fishbowl, which some people here were part of, including Jim. And The goal of Fish Club was to harmonize all of these bottom shelf surveys, which was like a Herculean effort, which we had no funding to do except to occasionally get together in working group meetings that were sadly mostly on Zoom. So it was funded by SAFS, which is a synthesis center in France. And we've done all the difficult work of bringing together all these surveys and standardizing the taxonomy, providing options for spatial temporal standardization, cleaning the data extensively. So now you can use them, and we'd love it if you did. We also need future funding to maintain and expand this database. So if anyone has ideas for funding this kind of infrastructure project, this is a challenge, let me know. But in the meantime, it's been a really fun way to pursue a bunch of different projects, not just the heatwave project, on global change impacts at a huge, huge spatial scale. This project, the Marine Heatwaves project, has the data set that we use is 28 million rows to give you an idea of how big it is. Okay, so let's see with all of those samples how biomass responds to marine heat waves. What I'm putting here on the x-axis is the intensity of the heat wave, which I can tell you extensively how we calculated because all of our reviewers have thoughts. And they were right. And on the y-axis is the log ratio of biomass, which is log biomass this year divided by biomass last year. It's a indicator of change. If it's positive, biomass went up. If it's negative, biomass went down.
And do you want to reiterate the no tweeting? Is that?
Oh yeah, thank you. So this paper is in revision, and while the rest of this work is all like open online and please tell everyone, these plots should probably not be generally shouted from the rooftops until we hear back what's on the paper. Thanks. It's a good note. Okay, so here's what happens. There is definitely no negative effect of marine heat waves. The positive effect that you see on the line of best fit is not significant. And I'll highlight on here, maybe so that you believe me, that there is no effect of marine heat waves on biomass. Some of the famous heat waves off to the right of the plot. So we've got Gulf of Alaska in 2017. I should note this is the survey in 2017, so it kind of followed the blob heat wave. And you do see a decline, but you also see the Northeast doing great in 2012, which makes sense. We know that in that heat wave year, the fisheries in New England had like one of the best years ever. And same in the Gulf of St. Lawrence. The Southeast survey is a small survey with a lot of variability, so that kind of explains why it's out here. And if you're wondering, okay, well, maybe it's not predicted by the intensity of a heat wave, but it is based on whether or not there was a heat wave at all. It is not. That's what these show. The distribution of biomass change is centered on zero, regardless of whether or not there was a heat wave. So that
Did you look at maybe abundance?
Instead of biomass.
Yeah, because the biomass has a lot of inertia. Big fish could probably weather the.
Yeah, that's cool. We did look at abundance. It's the same pattern, although I didn't know that, which makes sense. Some of these are missing abundance, so it's not quite as many surveys. I think Northeast and a couple other ones only have, no, not Northeast. A few of them only have biomass, but it was 15 or 16.
Cool. So I'm pinpointing something similar in the Bering Sea, and you don't really see a big change in the biomass with this last rating, but you do see very strong reactions in recruitment. Like
Oh, cool.
Super minimums, super maximums throughout that period of time. Things are diverging strongly.
Yeah, and
You pick that up with biomass.
no, we tested a lot of things, which I'll show you later, but that includes temporal lags, but they can't account, because it's such a large scale study, for differences in lags based on when the recruits would have shown up in the survey, which is I think what you need to do to detect that, which we couldn't really do. So we also tested this tropicalization hypothesis, and this plot is going to have this kind of weird thing on it, species thermal bias, which is calculated by, you look at a species and you look throughout its whole range and take the weighted average temperature, which is found. We call it the species thermal index. So that's its personal favorite temperature. Then you average those for all the species present in the region and ask, is it warmer or colder than the average? If it's warmer, it's a relatively warm affiliated species for that region. So it'd be over here. And if it's colder, it's over there. So it's relatively a colder affiliated species than what else you find there. What we thought would happen is that the cold affiliated species would lose biomass after a heat wave. Every point on the slide that I'm about to show you is a heat wave here, and that they would gain biomass after, if they were warm affiliated. Okay, awesome, basically on. And what we see instead is this beautiful cloud of points. There's no signal whatsoever, and there's also no relationship with the intensity of the heat wave, which is what I'm showing you with the colors. And when we aggregate these species level metrics into something called the community temperature index, which is sort of a single temperature value in degrees Celsius that summarizes how hot or cold affiliated the species present are. Similarly, there's no difference between that value when there wasn't a heat wave. So the last thing I wanted to do, which I can hold, was just to show you these plots kind of specifically for the Northeast Pacific and the blob. I'll hold those. If you want to come see them afterwards, you can find me, and kind of skip to our conclusions, which is, which are easy to say. There's no general effect on marine heat waves on biomass, and no general effect on tropicalization or community composition in the demersal fish that are caught in these bottom trawl surveys. We tested many things in responses to reviewers number 1 through 4, and I'm happy to explain any of these. It's been a fun few months. But we were sort of surprised that we really tried accounting for a lot of things that we genuinely thought would reveal kind of a decline effect or decline impact of marine heat waves, and none of them did. So curious for why, and happy to discuss with people.
Curious to, I mean, perhaps it's system specific, so some the systems reacts well, as
Yes.
you showed in the plots, some reacts bad, so facts that you don't see any like effects, it's perhaps that there is some like system effect where there would be a positive effect for some systems and some negative for others, so perhaps like aggregating all in one analysis.
We did
Okay.
do that. It's harder because you lose a lot of the statistical power, then you end up with like 30 data points instead of many hundreds, but none of them were significant. And even sort of qualitatively looking at them, you don't see places where there is a really believable trend, and it's just not significant. We also fitted models that just looked for changes in kind of absolute variability, regardless of whether it was an increase or decrease, just is there more change after heat waves? And that's also not the case. So that would have, if some places are losing biomass and some are gaining biomass, but there's always more change after heat waves, that would have revealed it. We didn't find that either. So, and if people need to go, you can go. I'm happy to keep chatting, but this is basically the last slide for the last part of the talk. So keep asking questions. But... Oh, let's get out of here.
So feel free to chat, but maybe out in the hall.
All right. Thanks. I'm gonna hop off.